The bacterial growth curve of a particular species of bacteria can be obtained. The growth curve of a population of a particular species can be obtained by growing a pure culture of the organism in a liquid medium at constant temperature. The samples are collected from the culture at fixed intervals every 30 minutes. After the number of viable organisms is determined, let's talk about the phases. The lag phase is also known as the adaptation period. During this phase, the bacterial cells start to adjust to their new conditions. The time period of the lag phase may vary based on how complex the environments are from the environments that the bacteria came from, as well as the condition of the bacteria cells themselves. If the growing cells are shifted from one type of media into the same type of media, which has the same environmental conditions, will have the shortest lag period. Those cells are damaged and will have a long lag period because they must repair themselves before they start reproduction. And the second exponential or lag phase, after accumulation of essential components which are required for the growth of the cells, they proceed into cell division, where one cell becomes two cells, becomes four, becomes eight, at iba pa. During this phase, the cells are divided steadily at constant rate. That way, the log of the numbers of cells plot against time result in a straight line. Besides, the population is more nearly uniform in terms of the chemical composition of cells, metabolic activity, and other physiological characteristics. And the third one, stationary phase. When the bacterial cells within a closed system, such as batch culture, run out of all the required nutrients, chemical, or their growth are inhibited by the waste products which is produced by them or lack of physical space, leading the cells to enter into the stationary phase, death phase, or decline phase. After the stationary phase, the bacterial cell enters into the death or decline phase, where the number of viable cells decrease in a predictable or exponential fashion. Long-term stationary phase. Long-term growth experiments reveal that after a period of exponential death, some microbes have a long period where the population size remains more or less constant. The phase is known as the long-term stationary phase. The bacterial growth curve is used when trying to utilize or inoculate known amounts of the bacterial isolate, such as to improve plant growth, enhance the biodegradation of toxic organics, or produce antibiotics or other natural products at an industrial scale.